Hi everyone, I'm Yun, PhD student of the Australian National Insect Collection of CSIRO and Australian National University. Today I would like to talk about our book project, the chapter of Cantharidae in Australian Beetles Volume 3. At first, let me introduce the family Cantharidae. They are also known as soldier beetles and belong to the superfamily Elateroidea. Cantharidae is a very diverse and is the 10th largest family of Coleoptera, including 5 subfamilies, over 120 extant genera, and over 5,600 species. In general, each subfamily and major tribe has the following distribution patterns. Cantharidae is concentrated in Holoarctic region and extends to Oriental area. Cine has global distribution. Demosphotheridae has Gondwanan distribution, including Africa, Australasia, and South America. Coleoptera has 2 tribes. Coleoptera is distributed in Australasia and America, and Ixirinae is widely distributed in tropical area. And Marcinae has dissimilar distribution with Cantharidae, including Holoarctic and the Oriental regions. Cantharidae beetles are soft-bodied terrestrial beetles with small to middle-sized length. Their adults usually visit flowers during spring to summer and can be easily collected by sweeping the vegetation. They are also opportunistic predators preying on other invertebrates, and some species can be attracted to the light. They have paired abdominal glands secreting the chemical defensive compounds when feeling threatened. Cantharidae larvae are also predatory and can be collected in the soil, leaf litter, or inside decaying wood. Some Cantharidae adults involved in lycidic mimicry complex. For example, the left side are lycidic beetles and the right side are Cantharidae beetles. They share the overall similarity, including the reddish color patterns, body shape, and the antennal type. Based on a recent phylogenomic study, Cantharidae originated from the late Jurassic. The fossil records of Cantharidae are also abundant, especially in Mesozoic Burmese amber and the Cenozoic Baltic amber. The molecular phylogenetic analysis of Coleoptera in Zhang et al. 2018 has revealed the possible relationship among 5 subfamilies. So now let's talk about the Australian fauna of Cantharidae. In Australia, there are 3 subfamilies, 3 genera, and nearly 120 named species. These species were mostly described from 1880s to 1920s. William John Macleay and Arthur Mills Lee contribute a lot for descriptions of Australian Cantharidae. All the named species of Australian Cantharidae are endemic to Australia. Among these Australian Cantharidae beetles, Coleoptera lugubris, also known as plague soldier beetles, may be the most famous Australian Cantharidae beetle. The reason they are called the plague soldier beetles by gardeners is because massive number of them appear on plants through spring, summer, and autumn. Although they look scary to gardeners, they are not really harmful for plants. Most of them were copulating or sucking nectar from flowering trees rather than eating the plants during the infestation. The interesting thing is, CSIRO researchers have recently announced the chemical secretion of this Coleoptera soldier beetle, and they found the genes that give the chemical its antimicrobial and anti-cancer properties. This may one day lead to a development of new antibiotic and anti-cancer related products. Now let's go through each group of Australian Cantharidae. The first one is Demosphotheridae. It contains 1 genus, Heteromastix, in Australia, and 77 named species. This genus is also distributed in New Guinea, although most of the species are documented from Australia. Most of the species were described by Lee during the 1900s to 1920s. Heteromastix is a very diverse group, and it is very likely that there are still many undescribed species in either Australia or New Guinea. For the Coleoptera, only nominal genus of Coleoptera is recorded from Australia, and 34 species were described. This genus presents surprising disjunct distribution between Australasia, including Australia and New Guinea and America. Most of species were described by McLeod during the 1870s to 1880s, and Lee during the 1900s to 1920s. And the same for Sydney. Only one genus, Symposium, is recorded from Australia and only 8 named species. This genus is mainly distributed in New Guinea and Southeastern Asia. Australian Symposium was mostly described by McLeod during 1880s. So what is our motivation for reviewing Australian Cantharidae? Maybe many of you might know that we are currently organizing a book series called Australian Beetles, such as American Beetles or Handbook of Zoology. We have already published Volume 1, including general introduction to each Australian beetle family and the key to all beetle families in Australia. For Volume 2 and 3, we want to provide detailed introduction to each family and the key to genus level for all Australian beetle family, which is the most important part. And I will be in charge of editing 3 beetle chapters, including Cantharidae in Volume 3. Additionally, I myself also want to contribute more or less to Australian Cantharidae fauna, as I'm a taxonomist of Cantharidae. Therefore, in this project, ideally, we will firstly do a literature review for Australian Cantharidae, summarizing their taxonomic history, biology, and the species checklist, and identify our collection specimens based on type specimens and the literature to see if there is any new genus or species or new synonyms, and describe those new whenever time permits. And finally, we can generate a key to genera of Australian Cantharidae. And this is our project diagram. Briefly, we combine the literature review and the specimen identification to see if there is anything needs revision or description and summarize them into our book chapter. Currently, we have summarized the checklist of Australian Cantharidae and the available images of type specimens based on the online database of LA. The first number is the type specimen over its images we assessed, and the latter is the total number of named species. We herein list the current changes in the taxonomic study of Australian Cantharidae. First, the type specimens are the keystone for species identification, and the type specimens are still the problem for us, as images of some species are unavailable. The name-bearing types of many species are syntypes, so the designation of lectotypes are necessary, and it will be possible that there are more than one species in a single syntype series. In addition, due to no Australian endemic genera, it will increase the difficulty in comparing those species distributed outside the Australia. For the generic classification, we have also found some issues. In heteromestics, we found some species have spindle-like maxillary palpomere, but some species have X-shaped palpomere. It suggests that another new genus may need to be established. For Coleoptera, Australasian and the American species might need to be separated into different genera. And the same for Symposium. Whether Australasian and the Asian species belong to the same genus remains unclear. Apart from the timetable, which may cost long for perfectly addressing the taxonomy of Australian Cantharidae, the major issue currently is the global crisis of COVID-19, preventing researchers from doing the international visiting, field works, and access the essential equipment from their office. Therefore, now we are still unsure how far we can reach our goal for Australian Cantharidae project before the deadline of the book project. I would like to express my thanks to my supervisor, Dr. Adam Simpinski, for inviting me to join this cool book project. Thank you for your listening.